Well, thanks for the introduction, and thanks for the invitation to come and present here today. Just a little bit about me, perhaps. I am living my dream. I absolutely have, I think, the best job in the world, at least those are my thoughts. It's a complex one on occasion. I actually have 50% of my appointment at UC San Diego at Scripps Institution of Oceanography. The other half is in the School of Pharmacy, and the sense of that, to be a professor of oceanography and pharmaceutical sciences, is we are engaged in drug discovery from the natural world. And so with my students, we go to places like Papua New Guinea and Madagascar and collect marine organisms, marine seaweeds, and other types of bacteria, bring them back to San Diego and engage in drug discovery research. That's one aspect. The other aspect, as passionate as I am about that, I'm equally passionate about the subject of today's talk. And that's using a lot of that same skill set in chemistry and algae and thinking about some other societal needs. And in this case, our need for energy, our need for liquid fuels for aviation, for all kinds of transportation now and I think into the long-term future. And so that's the subject I'll talk about today. So to get us started, I show the Keeling Curve. This is actually a professor at UCSD, now passed away some years back, but he discovered that there was a steady rise in atmospheric CO2, beginning with measurements back in around 1960 from Mauna Kea in Hawaii. And you can see the seasonal cycling of CO2 levels in the fine amplitude variations. But the general trend from 1960, this red line, the general trend is from lower levels that historically were close to pre-industrial revolution to increasing levels. And coincident with that is then starting around 1990, the observation that indeed this higher atmospheric CO2 was causing the dissolved CO2 in the world's oceans to increase. And the result of that, because when CO2 goes into water, it forms carbonic acid, is it was acidifying the water. And we have been able to actually measure in the world's oceans a measurable decrease from about 8.12 to about 8.07. So a significant acidification of the world's oceans. Think of the immensity of the oceans, and we've actually, as a species, we've affected the pH of all of that water. That's just staggering to think about. And so it's having major issues. The increase in CO2 in our atmosphere and in our water is having major impacts on climate and on biodiversity of the world's oceans. And putting this in a larger historical context, back pre-industrial revolution, we can see that the last years, as measured by Keeling and previous measurements, are taken from ice core measurements subsequently, that this is really a dramatic increase that's occurred in the last 50 years or so. And it's probably not going to get any better. As we look at our historical use of various fuel types, liquids, coal, natural gas, renewables, and nuclear, they've steadily increased, and we can predict that they're going to increase still into the future. So I think the key is that we really need to work on this renewable if we're going to pass along a healthy planet for the future generations. So there's generations of biofuels that have occurred. These are fuels that come from life forms that we can put in the gas tank or derive power from one source or another. So we have first generation corn ethanol, vegetable oils. These have advantages in that it's an established technology, but it's not very sustainable. You're interrupting or you're using land space that's actually currently in producing food. And so you have food and fuel competing with each other. There's fertilizer pollution. There's relatively low productivity from this activity. But it's what we have today in the Midwest and in countries like Brazil. There's extensive use of that type of biofuel. Second generation is this cellulosic ethanol. We're using wood chips. It does not compete with our food. which is positive, but the enzymes that engage in digestion of this raw material to something that then can be fermented and make a biofuel are very costly. Third generation represents green crude. That is the oil that we're going to generate from algae, and that's going to be the subject of the majority of my talk here. But just to summarize, it's using various types of algae. It does not compete with food. We are going to grow this in other types of environments that are not suitable for cultivation of food products. High productivity of algae uses CO2. Actually, all of these processes, you're growing as a result of absorbing CO2 out of the atmosphere, and so when you burn it, you're just cycling it. It's not putting new CO2 into the system, but it's actually quite costly at the present time, and I'll talk about some of the reasons for that. And we can envision in the future some engineered microbial biofuels. They're in the horizon, but are still going to require sugar. What's attractive here is we're using CO2 directly to produce the end fuel product. So that's very attractive. So diverse energy solutions, I think this is the key. So we have our biofuels, but also wind, solar, geothermal. Perhaps we'll see fusion energy at some distant point in the future as well. Now, looking at land use, if we think about the current first generation corn and soybean, if we're going to produce 50% of our current petroleum diesel use needs, to grow that in corn would take essentially the land mass of the United States, just completely impractical. The soybean is somewhat more efficient as the black box. Color somehow changed in the presentation, and so that's still not reasonable. But algae, because they grow so densely and they produce so much oil per unit space, you're not putting any energy into growing roots or stems. All of that energy is going into the types of cells that produce the oil that you want as the final product. So that would actually fit rather nicely in the state of New Mexico. And actually, surveys have been done by the Pacific Northwest National Laboratories of locations in the United States that would be suitable for growing algae. And you can see there's extensive locations in the south and the east coast for producing this type of product. And is this a dream? No, this is actually happening. Sapphire is a leader in this field. They are located in San Diego, so we have some connections with them, but no financial disclosures to worry about. But they have a production facility in New Mexico. You can see the size of these buildings in the foreground. This is an extensive facility, and so it's really occurring. They're producing green crude that they actually sell to then oil refineries that put it directly in the same system that they put the oil that they would pump out of the earth into. So some advantages is of biofuels from algae is that we can directly use photosynthetically fixed CO2, very high productivity as I talked about. We're not using arable land as I showed on the previous slide. This is not a nice agricultural land by any means, but it's perfect for growing algae. We can grow them in salt and brackish water, so we're not competing with our water supply. We can use, they have very effectively used nutrients in the water, and we can use wastewater nutrients to, in fact, stimulate their growth. And at the same time that we grow them for the oil, we can also produce some byproducts that help in the economic chain of actually making this a viable process. But at the same time, there's a number of problems that we have to overcome. For instance, we need to learn more about how to increase the productivity of these systems. As high as they are, that's in a laboratory. Translating that to these ponds is still difficult. And it's because we don't really understand the regulation of how they produce these lipids, these oils that we're ultimately going to burn. There's a major concern that here you're growing this pond of algae that's just anything that would eat it would just go in there and go crazy, having this meal of nice succulent fat cells of algae. So that's a concern, is that it's an open system that contaminants could come in. So how to deal with that in an environmentally safe and benign way. And the technology for harvesting all of these micro cells out of this big volume of water, that's a technical engineering challenge. And at the same time, there's opportunities, as I mentioned, for co-products like proteins or pigments that could be useful. So I'm going to talk, and I think in part the reason for my invitation to this event today is we recently published a paper in the Proceedings of the National Academy that talks about a new way to regulate, to increase the biosynthesis of lipids in these algae. And it's been really a phenomenal story, and a great graduate student. I'll tell you a little bit about that. So it begins with actually 4.5 years ago, a student, Trent Acosta, came to my laboratory at Scripps as a graduate student and had this passion for doing something in the biofuels area. And she developed an independent hypothesis, and it goes along the following lines, that this is a depiction in a very rough way cell, and basically we're talking about light and CO2 coming in, and by the process of photosynthesis, fixing this CO2 into sugars. And those sugars feed into various biochemical cycles. Some go into carbohydrates, and some go through more convoluted routes down to lipid droplets, or triacylglycerols, we call them TAG. And others in the field of microalgal biotechnology have thought about increasing lipids by maybe you could increase the production of this by cutting off the flow of carbon to carbohydrate, for instance, to knock this out using genetic engineering. And people have done that, and in fact it works. You in fact produce in an individual cell, you produce more of this desired product. But the problem is that every time that that's been done, there's been a coincident decrease in the growth rate of the cells. So you produce fewer cells. Each cell has more lipid, but the total that you get from, let's say, a flask is about the same. You really don't get much for that effort. Other people have approached this in the following way, and that is to increase the flow of carbon into triacylglycerol, so not knocking out carbohydrate biosynthesis, but just overexpressing some of these enzymes. And again, they've produced more lipid, but it seems to have a negative impact on growth. And so at the end of the day, you don't get anything more than the wild type organism. So Emily had the really quite ingenious idea, well, wait a minute, there's something also going on to these lipid droplets. They're being catabolized. What if we knock that out? And simple thought that it is, nobody had tried that. Nobody had actually thought about this before. And Emily took this on as her PhD thesis. Speaking of which, she defended yesterday. She's now Dr. Emily Trent Acosta. And so we had a great celebration yesterday afternoon. So she worked in a diatom. This is the diatom Thalassiosyra pseudonana. It's a very attractive model organism in which to do these kinds of studies because it is very productive. It produces a lot of oils just naturally, which accumulate as lipid droplets. It has a sequenced genome. That is, we've gone in, others, Mark Hildebrand's laboratory at Scripps and others went in and sequenced the entire stretch of DNA that makes up the genome of that organism. And that has enabled a number of molecular techniques that we can go in, we can knock out genes, or we can add in other features to this organism. And so that's what Emily did. And she did it as a result of another experiment that was done. It's been found that if you inhibit the growth of the diatom, if you starve it of some essential nutrient, diatoms have a cell wall of silica, glass basically. If you starve them of silicon so that the cells can't divide any longer, they accumulate oils. So again, there seems to be this correlation, lack of growth, increase of triacylglycerol oils. And so they went in and studied what are the genes doing during that period of time when it's accumulating oil, and found that some of the enzymes that are downregulated, coincident with increase of lipid oils, are some of these lipid catabolizing enzymes, lipases they're called. And so this is how she got the idea of what she wanted to knock out in this diatom in order to see if it would increase lipid production. And so she did that, and I borrowed this from Emily as a really very nice little graphic. This is a so-called plasmid. It's a design, it's a little stretch of DNA that you can insert into the cells and carry in some genetic information into the cells. And so we call that a plasmid. And the way that this experiment works is that she took this plasmid and she attached it to little tungsten particles. And then she put these tungsten particles into this machine called a gene gun, and it does pretty much that. It uses air pressure to accelerate these little tungsten particles into a petri dish that's depicted there. And so those particles are shot into the petri dish, into the cells. You have the diatom on the surface of that agar of that petri dish. And so the tungsten, some of them penetrate through the cell walls, do not kill the cells, but they carry that genetic information that she's built into the cells. Then you put the petri dish underneath a light, grow it for a period of time to let it recover, and then she had another selectable feature that allowed her to just isolate the cells that had incorporated this plasmid from the beginning. And so doing that, what did she find? Was her hypothesis correct? It was. In fact, these cells that have incorporated that new DNA, they show the same growth. This is days, or hours actually, and the number of cells. And the wild type is labeled WT, and the two transformants that she made this way have equivalent growth rates. So there was no impact on growth. So the big question is, did they accumulate more lipid? And you can guess, yes, they did. So this is, again, after silicon starvation, and we could do it also in conditions of replete media where it had all the silicon, but very dramatically here, here's the wild type accumulating lipids over this period of time of 72 hours. And the two transformants she made, they have about double the lipid. So this is a phenomenal effect, and it has no negative impacts on growth. This is the first time that that's been seen, and that's why we published it in that journal. I have another student that's working in this area, and just very briefly to finish the story today is an individual, Cameron Coates. He has done some very interesting work studying another class of algae called blue-green algae, or also known as cyanobacteria. We study these for many reasons, including some of the very interesting chemistries. This next week I'll be up in San Francisco with a company that's trying to develop one of our compounds as an anti-cancer cure. So we're finding some really exciting things from these cyanobacteria. But they also make these very simple compounds. These are linear alkanes. They're not unlike if you have a diesel vehicle, you filled up your tank perhaps this morning with diesel fuel. You filled it up with things that look very similar to these molecules that are being made by the cyanobacterium. We really don't know why cyanobacteria make them. We didn't really know going in if all cyanobacteria do make them, and do they make the same ones or different ones, or how are they made? And so there's a lot of unknowns here, and this has been the subject of Cameron's thesis. And what he found, so a lot of detail on this slide, clearly you can't see it, but lots of different species of cyanobacteria that Cameron grew in the laboratory and analyzed very carefully. Very nice work, a beautiful piece of work to quantify the amount of these hydrocarbons that are present in different cyanobacteria and structurally define them with great rigor. And that paper's also just come out in the PLOS One journal, which is, again, a very nice journal for this kind of publication. And what he found is that every cyanobacterial strain that we've studied makes these hydrocarbons, and we still don't really know what their function is. So that's very interesting. That they're made by two different pathways is very intriguing. So the capacity to make them has emerged more than once in the lineage of cyanobacteria, which there's a lot of interesting questions about that. And what we found, I think, most interesting is that there's a subset of these algae that he studied that actually make branched hydrocarbons. So branched hydrocarbons have, let's see, we'll just go back. They have branches like this methyl group here. Why is that interesting? Well, that's interesting because these are the features of, if you didn't have a diesel engine, but rather a gasoline engine, you filled up your tank with branched hydrocarbons. So gasoline has different burning properties and fuel properties than diesel fuel, and it's the nature of the branching carbon atoms. So this is very exciting to us because we're now in a position of understanding how those branches are introduced. And I have a third student working on this topic in the laboratory right now who's trying to entrap those enzymes that are making those extra branch methyl groups and have algae actually create gasoline directly. So you would be harvesting CO2 out of the air and with sunlight and water and directly producing gasoline. I think that's just a a fabulous idea. I don't know if we'll be successful, but we're going to have some fun trying. Okay, so just some conclusions here. I feel our planet is in peril. I think that's the right terminology to use. We have severely impacted our planet, planetarily, worldwide, in terms of the increase in CO2 in our atmosphere, which is causing climate change issues, and increase of carbonic acid, which is CO2 in our world's oceans, which has created ocean acidification. This is a major concern. For instance, calcium carbonate organisms that have protective shells, like mollusks or corals, they're very dependent, very sensitive to changes in pH as to whether that calcium carbonate is going to aggregate and form a shell, or is it going to disaggregate and dissolve. We're at a tipping point for some species that we're going to actually start to see things dissolve rather than their enzymology be able to aggregate calcium carbonate. So this is major concern. I think that we'll meet our future energy needs from a diversity of sources, and I think that algae is a part of that. Let me just reiterate, this is a carbon neutral source. When we pump oil out of the earth, this is ancient carbon that's been buried there for millions and millions of years. That, when we take it out and rapidly burn it, we're adding new carbon into the atmosphere. But when we take carbon out of the atmosphere, fix it into lipids or hydrocarbons that the algae make, and then we burn it again, we're just cycling that CO2. So I think that's where we should be really setting our future sights on, is this kind of a sustainable type of fuel. And increasingly, let's see, you were talking about electric cars just a minute ago. I think we'll be seeing more and more electric transportation methods, electric boats and the likes. But if we're going to fly an airplane through the air, I don't know that there's any way that anybody's conceived of to power that without having a liquid fuel. So I think our dependency on liquid fuels will be a long-term, and for a long time we'll have a need for them. And I think that algae will provide a source for that. But we need to overcome a number of technological challenges, as I've mentioned, and I think we're making some progress through the work of students like Emily and Cameron. And I'd like to just finish on this note that Scripps and San Diego area is really showing some leadership. I'm proud to be a part of this. Steve Mayfield has pulled this together. He's quite a famous scientist on the UCSD campus. Has pulled together and created this California Center for Algal Biotechnology. And it's quite an impressive center and an impressive group of colleagues that have pulled together that have some interest in algae, from the basics of the photosynthetic process to the engineering of how one might harvest or grow these organisms more efficiently, to people such as myself that are studying their chemistry and biochemistry. With that, I thank you for your attention and look forward if we have any time for questions. Thanks very much.
Thanks, Bill.